Always please report on like the noise once again. Students once again in the hallway, please report to the multi-purpose room.
All right, guys, we are a 46. We are going to be doing our cell membrane foldable today. And it is the 29th of January. So our mindfulness minutes is called Find Your Focus, and it is from Headspace.
Sitting comfortably, just beginning with a nice big deep breath, breathing in through the nose and out through the mouth. Your eyes open, a soft focus. You breathe out, you can allow the breath to return to its natural rhythm. Just keep the eyes open for a moment, focusing in on one particular object within your field of vision. Staring at it too intently, still aware of the space around you. Gently focusing on that one object. Slowly zooming back out again as you blink a few times with the eyes.
All right, guys, how are we feeling today? How are we feeling today? Go ahead and get in. And if I double check my sign-in sheet, I have Marley, professional computer thing. Annual. Hey guys, so if we go ahead and take a look, I've got a whole lot of you that are tired, a bunch that are, looks like more warm than cold. We are almost exactly at 70 degrees. We also have a lot of hungries, a little bit of boredom, some optimism, some joy, ecstasy, some love, a couple of acceptance. Mention a terror. Terror feels really intense. We're here at the school day. That one jumping, some other optimism there. All right, guys, I do want to remind you a couple of the general points here for the Pear Deck. So every day we are supposed to be in uniform. Guys, those ID cards should be around your neck. When admin comes through to do the checks, they want to see the ID around your neck. Do not have them all the way put away in your bag. So please make sure they are not all the way around in the bag so that when admin pops into the room, we can easily have them. Also, we are not supposed to have any hoods. As my hoods need to come down, please. We're not supposed to have our hoods on our bags. Also, we are not supposed to have any solid backpacks. If your clear backpack from the beginning had gotten messed up, you can pick them up from the front of the school is where they pass those out. Pear decks, the pear decks from the 17th to the 19th will be closing on February 9th. That's not this Friday, but the following Friday. The pear decks from last week will close the week after that on the 26th. And the ones from this week will be closing on February 23rd. Are we good with this idea? That gives you kind of like a 3-week run. All right, here is where we are in our sequencing. We are going to do our cell membrane foldable today. Next class, we are talking about osmosis and diffusion, and we're going to do our cell quiz corrections. And then we have our membrane reading on Friday. And this is what it looks like on the calendar. All right, so thinking back to last class period, what is the difference between diffusion and facilitated diffusion? What was the difference there?
Facilitated diffusion can.
Yeah, it's got to go through a channel. It needs to find a door. That is really at the core of the difference. Diffusion and facilitated diffusion. It's high concentrated to low. It's stuff spreading out. The only difference is if we have facilitated diffusion, it has to find a doorway. It has to find a channel to go through. It just can't evenly spread out in every direction because there's cell membranes or walls in the way. Yes. But not of every molecule, right? Some molecules can just spread out and they can go through the cell membrane without a problem. Other ones can't. So for example, could water just go through the cell membrane? No, that's why we have the cell membrane there, right? Keep our water separate from their water. But oxygen is able to pass through without a problem. Does that kind of make sense? Because oxygen's not polar. So the lipids don't do a great job of keeping oxygen. So the oxygen just spreads out evenly. The water would have to find a channel. Good. An analogy here is if the classroom was a cell, we would have to be like our facilitated diffusion, right? We'd have to find a doorway to get through. But again, things like oxygen, even if we shut the door and we have the windows down and we close all the doors, would we start to run out of oxygen? You think this room is airtight? No. You would have diffusion under and around the door frame, right? Through the ceiling, like all up there is not like separate room to room. Right? It's that drops it like there's open space up there that has all the wires and pipes and everything else through it. Air would come through. Right? It would not be like you wouldn't have to be like, oh no, we're gonna run out of oxygen. That wouldn't happen to you. In fact, it takes an incredible amount of tech to try and get a room to be a vacuum seal. In fact, it is extraordinarily expensive. So when you go up to like NIH, if you are working with infectious disease, they do have some vacuum locked rooms, where you have to go through like a decontamination process so that like really nasty pathogens don't get out in the populace. But we're talking like a couple $1 million to put one in place. Pretty much every one that you would normally see is not actually here. Yes, but the amount of time it would take you, even in a perfectly air-sealed room, you wouldn't run out of oxygen. Like it would take an incredible amount of time. No, just regular diffusion. Facilitate, think of facilitated need like you need a facilitator, something to help. That's where we need a channel to help the things get through. Okay. Does that make sense? Where you have like, even when you have like the people who've like asphyxiated, etc., like, oh, they were like in a mining cave-in or something like that, they didn't run out of oxygen. That's not what happens. What happens is they have too much carbon dioxide buildup. And that's what kills people. It's not running out of oxygen, it is the carbon dioxide. So like, even if like where you will sometimes see like, oh no, they ran out of oxygen is primarily diving accidents. Where there's not any air, and you brought the oxygen down with you, like you had that big tank. But most of like, even when you have like scuba divers and stuff, that big tank isn't oxygen, it's compressed air. It's just our regular atmospheric air, which is 78% nitrogen. Like that's what you breathe. You breathe nitrogen. 16% of our air is oxygen. You could breathe the same breath in and out 30 or 40 times before you got all the oxygen out. Like we don't, oxygen is almost never a limiting factor unless you are in water. What the issue is if you had like, let's say a plastic bag and you're breathing in and out, it's not that you ran out of oxygen, it's that you got carbon dioxide poisoning. Too much carbon dioxide built up and you weren't able to get rid of it. Does that make sense? Like a lot of times in movies and stuff, oh no, they ran out of oxygen, they suffocated. The suffocation was not because not enough oxygen, it was too much carbon dioxide. The carbon dioxide wasn't able to get on. That makes sense?
Yeah.
Yeah.
I have a theory. If someone like just took in like 2% of their off, but they would be able to output.
No, our body makes the carbon dioxide.
I know, but if they took in oxygen,
Right.
that's like oxygen.
No, that's actually what when you have the people who are on oxygen, sometimes they have to be on only oxygen.
I know, but it's like 100%.
Yeah. That's just ups the concentration. So if your lungs have basically failed and they're only working in like 2 or 3% capacity, they will fill your lungs with pure oxygen. You still have to breathe out your carbon dioxide. Your carbon dioxide came from the food you ate. So you don't like, we don't want, like we take in our oxygen by breathing, but the carbon dioxide we breathe out is not what we breathed in. Like we're breathing out the waste products from our bloodstream and our cells from breaking down our food. That makes sense? Okay. The oxygen ends up in water and you end up peeing it out. Like you breathe in the oxygen and you pee it out in water as your excess. The food you eat comes in as solid and we breathe it out as oxygen. Or breathe it out as carbon dioxide.
If you like swallow like too much air after eating, can it come out like as like a burp?
Yes.
Also kind of oxygen.
It wouldn't be as oxygen, it just basically, it would come out as mostly nitrogen. What you breathe, what we're breathing right now is nitrogen gas. With a little bit of oxygen. The difference is the regular atmosphere has 0.03% carbon dioxide. Yes, it's a tiny amount. The reason why we say, oh no, like the carbon dioxide percentages are going up is every tiny increment that carbon dioxide goes up gets more dangerous for us and it causes a lot of other things. Does that make sense? It's one of those substances that's like, we don't need a lot of it to have really big impacts. Like it tends to run fairly not great for us. Are we good with this idea? All right. Oh, if we look at the types of transport, if we have a cell membrane, so we have a cell, how are we going to get things in and out? So first we have diffusion. Takes no energy, it goes in any direction, and it goes from a high concentration to low, meaning it's spreading out. It went from where it's really packed in tight, trying to spread out. Are we good? We have our facilitated diffusion. Again, no energy, but this time it's only through channels, but we're still going out. The last type of transport to go through a cell membrane is what we refer to as active transport. Now, active transport needs energy. We got to put some energy into it. It does only happen through channels, but it goes from low to high. Guys, this is concentrating. We're taking it from being spread out and packing it in tight. Guys, this would be like when your parents say you have family coming over and your room needs to be cleaned up and you have to put all that energy into it to get it from being spread out all over your room to having all of the clothes folded and neatly put in the drawers, all the papers organized, everything put back where it's supposed to be, right? Is that no energy? Getting your rooms all cleaned up, does that take no energy? Right? The cleaner it has to be, what happens to the amount of energy you need? Or it goes up. Does this make sense? So when we want something to be super concentrated, where all of one thing is only in one spot, we're gonna have to put a ton of energy into it. If we just need it kind of sorted, but we don't need quite as much. Does this kind of make sense? All right, yeah. So it's not the cell membrane opening up. The cell membrane actually has a protein channel that makes a little opening. Kind of like if the walls and ceiling and the floor was the cell membrane, the door would be a channel. Like that metal around the door would be a channel. It can be open, it can be closed, but it exists as an opening in that cell membrane that things can go in and out of without like breaking the wall. Well, not because it's polar, it's literally a protein tube. Like, so sometimes we need to open and close it, sometimes we just leave it open. Right? So if it's something that if it's just for facilitated diffusion, I'm gonna leave it just open. Okay? So like the straws that are just open, sometimes I want to leave, right? So I might have a lock on it where it's like, all right, I don't need you open right now, so I'm gonna shut you. Kind of like we close the door. Does that make sense?
Yeah, but what is that?
Well, because we're concentrating. So we're not letting things just spread out. We're saying, no, I need you in here. I gotta take energy to pull it in. Because the natural state of things is it wants to spread out. Does that make sense? And so active transport is anytime our cell is like, for example, glucose. I need that glucose. Everybody else is trying to get the glucose. The cell, if any glucose goes past, literally is like, I need this glucose here, but it's gonna have to expend some energy to attract the glucose and pull it in when there's not a lot of glucose out there. Because the glucose wants to just spread out. So we pump it in to keep it concentrated. Okay. All right. Last class, we should have finished up our data and statistics assignment. Was there anybody who had any questions on our data and statistics? We're all good with that one? Do you want to also remind you guys that for your inquiry project, this one is going to be where you do just like we did with data and statistics, but it is going to be for your experiments. So you do want to start running those experiments, collecting your data. Guys, you should have at the very least 3 data points per group. At the very least. This is where I was suggesting, like if you're using seeds, do them in sets 10, right? If you're running trials, I personally always recommend doing like, if you're like, hey, can I practice with something or I'm counting something off a video, find 10. Because guess what? It makes your life so much easier when you go to do your calculations. If everything's on a base 10, it's like multiplying and dividing by 10 is a lot. Figuring out your percentages when you have out of 10 is a lot easier. Okay? Yes.
What if you like data that's like not like numerical?
So you will have to quantify some of it. So you can quantify things by assigning numbers, right? The pH scale is a good example of that when we do, I have one, I don't, I think I put them all away. Like when we do the colors and then they have the numbers associated with the different colors. So like you can do that. You just need to come up with like, hey, I'm gonna quantify these. If I put them on a spectrum of all of this way or all of that way, how would you kind of quantify it? Does that make sense?
Yeah.
And you can do that after the fact. And so after you collected all your data and then kind of put them on the spectrum and be like, okay, this is the most extreme this way, this is the least. You do want to, in your procedures, you want to add that step of like, how did you make your scale? Right? But yeah, you're allowed to do that. Guys, if you change anything as you're doing your experiment, you're like, whoa, that didn't work, I'm gonna do it this way. Make sure you change it in your steps, right? Because the initial procedures wasn't like a final draft. It was just, yes, that looks like it would work. If you found it didn't work or you couldn't find certain materials, like you do want to have your procedure be what you actually did, not just what you were planning on doing. Are we good? Anybody have any other questions there? All right, so as we go through, and we talk about and we talk about our cell membrane, okay? When we talk about our cell membrane, we have a couple of things. So first off, our cell membrane is primarily made of phospholipids. Now, the reason why they're called phospholipids, remember, us scientists, we are not actually creative when it comes to naming things. We describe what it is and call it its name. So our phospholipid has our lipid right here. Remember our glycerol and our fatty acids, just like we did on our macromolecules foldable. A phospholipid has a phosphate attached to the end. So that's why we call it a phospholipid. It's a phospho and a lipid. And that was how we named it. Now, guys, phosphates will connect to polar groups. Guys, where did you also see that word polar? When we talked about what other molecule? Water, right? So water was polar, which meant that it had charges. Lipids, are they normally polar? No, they have no charges, right? That's why they don't get along with water and they don't interact with water, right? Like oil and water don't mix. Fat and water don't mix. Our lipids and water don't mix. But we have this phosphate that allows it to be in water without just totally repelling. So they're called phospholipids because they have the phosphate. So what property of lipids makes it a good substance? A membrane. Remember, our membrane's main function is to be a barrier. What is it about lipids that makes it a good barrier in our body? Yeah, it doesn't interact with water, right? Do you want to go take a shower and dissolve down the drain? Right? Like we're not in that, was the elements, right? We're not like washing down the drain. That would be ridiculous. We want the water that's inside of us to stay. Oh, that's our water. That's us. We don't want to be like go to shake somebody's hand and just have it loop together. Like, no. We want it separate. The thing that keeps water separate is the stuff that water can't dissolve, which is lipids. Water dissolves pretty much everything else. Water dissolves metal and rocks. The only dissolved are our fats. Okay? Yeah. Yes. For phospholipid, the lipid part won't interact with the water. Having that phosphate on the end of it is going to let it actually form these nice flat structures instead of just making a blob. Like if I put oil in water, is it going to form a nice thin sheet? No, it like blobs up in the center. What that phosphate head does is it lets it like make a thin layer instead of it blobbing together. That the phosphate's going to face outward towards the water. The phosphate head would attract water. And then the tails, right? They're gonna be facing inward away from the water because they don't want to mix with the water. And so what we end up with is a thin layer that the water can't get through. It would have to go through a channel.
That would-
Right.
So usually around every cell there's more.
Yes, if a cell doesn't have any water around it, we end up getting dehydrated. Now we can exist like sitting out here with no water, right? But we still have to drink water in order to replenish ourselves. But the reason why we're not just immediately drying out is we have that layer of lipids not letting our water evaporate away. Does that make sense? But this is why if you've ever gotten like a cut, or a blister, right, where the skin, that lipid layer has been breached. Think about how your skin feels around it if it's not, like, that's why we put the Band-Aids on with the ointments to help keep it moist. That skin gets really hard and dry and brittle. So we have the scabs which are a little bit different. That's one that our body actually makes, but just the actual skin around, when you have like a blister, it gets like real like crackly. Like it's real dry. If you don't keep it with the antibiotic ointment and the Band-Aids on top. And the reason for that is you breach that cell membrane and so the water is able to evaporate. And so you get like this dry layer. Does that make sense?
What is it again?
So what do lipids not like to mix with? Water. So the property of lipids that makes it a really good membrane is the fact that it doesn't want to mix with water. It's like, no, I'm gonna pass. All right, so now if we look at these, we have the phosphate, which we refer to as the head of the phospholipid, because, well, it kind of makes this little circle, right? And then we have, these are the tails. They're the fatty acids. So our phosphate head is hydrophilic. Hydro means what?
Water.
And philic? Philo? Well, Philadelphia used this exact root word. What is Philadelphia's name? It's the city of. Huh? Philadelphia is the city of brotherly love. That is their like literally direct translation of Philadelphia. So phila means to like. Okay. So hydrophilic, it means it likes the water. It's good with water being around it. It's not repelling it, it's being there. Are we good? So that's this part here. It's attracted to the water. Our fatty acid tails are hydrophobic. Phobic meaning what? If they're afraid of it, they are actively avoiding it. They do not want to be near that water at all. Right? So down here is repelled by the water. So if you look down here at this picture, this is what our cell membranes end up looking like. Is we have a row of phospholipids on one side with the heads facing out and the tails facing in, and a row on the other side. Now the blue here is the water. Anytime I put phospholipids in water, it's going to form this layer. I can make them synthetic. I throw them in water. I get a double layer like that. Why would it do that? Yeah, so the phosphates basically go to protect the lipids. If I have right, another loose phospholipid that comes in, and it comes in like this. Is it going to be okay like that? No. Water? No. The water, they're gonna flip back around so that they're not. If I peeled off this entire top layer of phospholipids, would all of these down here be okay? No, in fact, if they were near another phospholipid, what they're probably gonna do is like fold themselves up and you just end up with a shorter double layer. Because those tails are gonna be like, get that water away from me. I don't want it anywhere near me. I don't want it touching me. Right? And the hydrophilic heads are like, okay, that's fine. Like, I'm fine with it. It doesn't bother me none. We're good. So what this means is anytime we have those phospholipids, we always get a nice, neat double layer. Are we good?
So that's it, like, what is up? It's always like.
Yeah. So anytime I put phospholipids down, they're always going to form this nice double layer.
I know, but some the inside is
Cytoplasm is 70% water. So it's going to be the water here, and the outside is going to be water. So they're always going to flip around to the inside. Well, it does, but only by the phosphate heads of the phospholipids. Those tails don't want to touch the cytoplasm. The cytoplasm has too much water. Does that make sense? So it's not pure water, but remember, we're 70% water. Every one of our cells is 70% water. It like the water is everywhere. And so the only way that these are happy is if they flip around and they're not like, they're like, okay, we're going to put our waterproof part on the outside. Like, let me just like keep that water away. We're good? Okay. Now, we have proteins just attached to one side. We call those peripheral proteins. That's like these ones here, right? Like the cabinets on the wall. Yeah. Because those tails are always going to flip around to face each other. They are always going to try and avoid the water. So you're always going to get a double layer. You're never going to see them like sticking like with just a single line of them, because then the tails would be touching water. Now, most of these are identity markers, right? So we have these surface proteins. There are identity markers. We also have transmembrane proteins that go all the way through. Now, we have transport transmembrane, which regulate the movement of stuff, and receptor protein that sets off a response. So guys, the door would be a transport, right? We can walk all the way through it. The window would be like our receptor protein. Do we know what's happening out on the parking lot? Can we see what's happening if something's popping off on the bus lot? Yeah, we would get noise, we get light, we know what the weather is, we get information. Does that mean all of that weather is coming inside? No, it just like it goes through the wall. We're just getting information through. We're not getting stuff. Does that make sense? That's the difference there. Transport lets actual things through. Receptor ones is just information of like what's out there, what's going on. Give me that information. Now, why would we need proteins that go through the whole membrane? Yeah, to open it up, right? Imagine we were in the room and there were no doors, no windows, no nothing. Would we be able to just hang out here forever? No, like we have to be able to get the stuff we need in, and we have to be able to like let stuff out. Like we can't just have it have nothing in and out. We have to have that ability. Yeah. How the proteins open and close? Well, so the peripheral ones are identification. So they aren't giving us messages. This would be like the sign out front that says that it's room 509 in Vegas classroom. That's a peripheral protein. It's attached. It lets people know what's in here. The cell doesn't necessarily need to have those, right? However, if you have an immune system we have to have the identification so that your immune system doesn't then kill your own cells off, right? So those are just the ID. They're like, hey, this is what happens here. The other thing that often happens on peripheral proteins is this is where you can have like signals attached. This is different peripheral proteins, like you can have two pieces on two different cells that lock the cells together so that the cells don't pull apart. We definitely need that on things like our epidermis, like our skin cells. We don't want our two skin cells to be able to be like, I rub my finger over it and I'm able to like pull the cells apart. I want them to stay stuck like Velcro, right? That's peripheral proteins. So then it would be either a transport or a receptor protein that we would put into our cell membrane. Our peripheral proteins just stay on the outside. They don't interact with the inside. They just hang out on the outside. Does that make sense? And think of it as almost like some of them are like the Velcro to hold them together next to its neighbor. Some of them are just identification of like, hey, this is what's here. This happens to be a muscle cell. Like, I am part of you. Please, immune system, don't go and kill me. Are we good? Okay. All right, so we are going to start, maybe finish our cell membrane foldable. If you had these from last time, if you could pull them out, otherwise I'm going to pass them out. I think most of you guys gave them back. What? Well, I didn't pass them out at all? Thought it was this class. I passed them out in one class and you just didn't get to them last class. I collected a setback. Yes. Because all of your foldables are your study guide for the is our state panel. All right, guys, so we are going to do the top part. Going to have us fill it in, and then we will color. Now, if you look at the diagram, I have diagrams. A top one that's going to have all of our structure and the protein. It's going to deal with our transport of how we actually move molecules across. Everybody have foldable paper? All right, so the first thing we are going is on the top, and I put it in the hair deck as well here. So our phospholipid bilayer. If we look at A, down here in this diagram, it has that little circle and the two little tails. What was that called? We just went over it. It's a phospholipid. So that little circle and two tails, that is a phospholipid. So A is a phospholipid. Yes. Good. And what do we use the phospholipids for? The main barrier part. Now, I have a diagram of a zoomed in phospholipid there, where we have the polar and non-polar label. The polar circle part is called the head. And the little non-polar tail, those are our tails. So, in that first block, we should have A is our phospholipid. Second line is used for a barrier. The next line down, where we have the bracket for the circle, is head. And then the last line at the bottom with the bracket on the non-polar parts, those are our tails. Now, if we go over a block, we're gonna look at our integral proteins. These are proteins that are in our cell membrane. So this was our B and our C in the diagram here. So B is what we refer to as a channel protein. So B is a channel protein. And what the channel protein is used for is letting things in, letting stuff in. So when that protein is built by the ribosome, well, actually for the channel proteins, usually it's the ribosomes on the endoplasmic reticulum because it's a little bit more complex. So it has a couple of pieces that fit together. It gets built and then literally inserted into the cell membrane. And when it's inserted as a tube. Yes. Now it's not rigid, but it does stay going all the way through. So it's kind of like a layer, but that layer can shift around a little bit. Nope, they leave it right in there. Now, if they want to block a channel, we'll get a protein that literally acts almost like a cork. And they just say, nope, we don't need you to be open right now. And they just kind of cork it. Some of them, like if you look down at the bottom one, when we talk about active transport, see how it's all like pinched shut? Down at the very bottom above active transport. That is also a channel protein where unless you have a certain receptor where an ATP has fit in, basically when you have, remember when we talked about how enzymes, when you have the substrate fit in, the enzyme kind of shifts shape. For our protein channels, if it's something like active transport, where we want to keep it concentrated and not let it just run back out, is we literally have, if you look here where that ATP fits in, it causes it to change shape and let a few in, and then it closes back up. Almost like those rubber coins first ones, like if you press it open, it opens, but if you stop putting energy into it, it like snaps shut again. Like this one. Find them from like the fair and stuff, they pass them out, I think. Those like little rubber like comfort mount. Yes. No, they are part of the cell membrane organelle. Does that make sense? So like we didn't go into like the pieces of the cell membrane. For the cell membrane, we just said, oh, it's the cell membrane, that was it. We're gonna zoom in a little bit on the cell membrane because in order to go over how they get in and out of the cell membrane, knowing what the structures is important. Does that make sense? All right, so C was our signal protein, and it lets the signals in. All right, that was B and C. Now, if I go over to the next one, we have these two structures that have these branch on top. Okay, that was our D and our E. Now, guys, all of those little, I know they're circles. Technically, I should have drawn them as hexagons. I did not. But it's these little tiny circles there on top, and that is glycogen. That is glucoses in a certain pattern. What that does is it lets them be an identification. Remember I said scientists were lazy at naming things? So we're going to put glyco as the prefix. D is a glycoprotein because the glucose was attached to a protein. So D is a glycoprotein, which just means a protein with some glucose. And E is one of those phospholipids that got glucose attached to it. So we're on a glycolipid because we attach the glucose to a lipid. Now, both the glycoprotein and the glycolipid, it is used for identification. Okay, the difference here is the sign I have on the outside of my door up above where it's attached to the wall. Like our glycolipid is attached to the wall. If our wall is like our cell membrane, that works. The orange one that has the room number on the glass of the door, if the door is like our protein, that's like our glycoprotein. It's just where did they put the sign? I have a sign both on my door and on the wall. That's really the only difference. It does not impact where the identification is. We just have a special name for like, hey, it's glucose, and what is it attached to? If it's attached to the lipid, we're going to call it a glycolipid. If it's attached to the protein, we call it a glycoprotein. We good there? All right. Our last two across the top was F and G. So F is simply a peripheral protein. It is P-E-R-I-T-H-E-R-A-L. And guys, what the peripheral proteins are used for is identification and messaging. Now, guys, if we look at that peripheral protein, do you think it's getting any messages inside of the cell? Is F going to the inside? Just hanging out outside. So it is messaging, but it's not to receive messages. We will see peripheral proteins sending messages only. Does this make sense? So it might be a warning off. It might be just, hey, we have this here. This is when I don't need to get any information back. I'm just putting the information out. The analogy I'd have here is like our PA system. Do we get to interact back with the PA system? No. But they give us messages all the time, right? They're like those peripheral proteins. They put the information out so that other cells, if they bump into this cell, can get that information, but there's not a back and forth on it. Our last one is G. G are these little zigzaggy structures inside where the tails are, and this is cholesterol. And what cholesterol is used for is to stabilize the cell membrane. It's given as support. Now, guys, cholesterol is rigid. Our cell membrane think of cell membrane as like vegetable oil, right? Does vegetable oil have a lot of structure to it? No, it just kind of flows and it's just kind of goopy, right? The cholesterol in our cell membrane is more like refrigerated butter, okay? It's got some structure to it. It's still a lipid, but our cell membrane, depending where that cell membrane is for what kind of cell, we want more or less cholesterol. If we want something to be able to be super bendy and flexible, not a lot of cholesterol. If we want the tissue to be more rigid, lots of cholesterol. Does this kind of make sense? Good with this idea? Yeah. Which? So the peripheral protein is for messaging and identification, but it's out only. So it's not a two-way communication. It's just, hey, I'm putting these posters on the outside so that the people out there know. You know, the big signs outside of a store being like, yeah, you have to have shoes to come in.
What about the F?
So on the inside, it would be like having cabinets where we might put stuff up as messaging within the cell, but it's not leaving the cell. So like our cabinets, so like I might put signs up to remind you of stuff when you're in the classroom, but I don't need everybody in the hallway to know. I can still put those messages up, but it's just put internally in the cell. That makes sense?
After the second, where it's inside of the cell.
Where that's zigzaggy, that's G.
No, after the second,
Second F at the bottom.
there's two F, one at the top.
Right, one's outside of the cell, one's inside of the cell.
They're saying that
They don't communicate with the other side. So it's like a sign I put up inside the room that doesn't, like the two F's wouldn't communicate with each other. It's just information cut out. But it's not like, so if I put message boards up here, right? If I put announcements up inside the room, does anybody in the hallway need to see it? It just goes up here. Same way as you might have flyers and stuff outside that we can't see inside. Cause that just can stay out there. Does that make sense? Yeah. Yeah. All right, so now guys, sometimes you'll hear the idea of like cholesterol being an issue, right? People have high cholesterol. Now, guys, if you had no cholesterol, you would die. It would be really bad for you. We do not need to worry about that here in modern America. Like, we get so much cholesterol that like we really do not ever have to worry about like, oh no, our body does not have enough cholesterol. It can't hold like rigid shapes of things of like our internal organs. Instead, pretty much everything we eat ends up with cholesterol. We've got so much cholesterol and so much salt that like we have to pay attention to get less. Now, guys, how many of you have ever heard when they talk about like medical stuff, somebody may be talking about hardening of the arteries?
Yeah.
Right? And where high cholesterol is often linked to heart disease. So, guys, what's happening there is that cholesterol, right? It's lipid soluble. It wants to join lipids. Well, when we eat it and we get a lot of cholesterol, how do you think the cholesterol we eat gets around to the cells? What does it have to travel through? Well, eventually the membrane, but what else does it have to get from our stomach and intestines out to the rest of our body? What carries everything? Our bloodstream, right? And when it goes through our bloodstream, it also goes through our heart. Well, guess what? If we have too much cholesterol, our arteries and veins are the first ones to start absorbing that extra cholesterol. It starts absorbing into the cells around the surface of our heart and our veins. Guys, do you think you want your blood vessels to get stiff?
No.
No. How many of you guys have ever had those straws of the like compostable plastic? Like they're the newer straws. They have this kind of odd texture. They're like the plastic that's supposed to be like better for the environment, but they like if you bend them at all, they like shatter and they crack really easily.
Yeah.
Like a lot of restaurants have started using them and it's irksome. I really hate those straws. I'd rather have no straw than those. But that's what starts happening to blood vessels. If you have your arteries are hardening, when your heart's pumping, right, it's gonna have like pressure and bending. And what can happen is the cells on those arteries actually can end up cracking because it's too rigid, right? It's too stiff. And so when it bends, we get little cracks. When you get the little cracks, you get blood clots that lead to heart attacks and stroke. Does this make sense? So this is why when doctors say, hey, you need to cut your cholesterol down, that's why. Now, it doesn't mean you can't ever have cholesterol. Like, we have to have it, right? We want to be able to have some of our tissues be more rigid than other tissues. And we do that with the cholesterol to help make it more stiff. But we don't want too much, right? And in our modern diet, that's usually the way that most of our diets tip to. Now, where you run into if you didn't is, let's say you went fully vegan, right? No animal products whatsoever. At that point, you actually have to be very conscientious about getting enough cholesterol because pretty much most animal products have some cholesterol in them, even if it's just like dairy, milk, butter, things like that. But if you have somebody who's fully vegan, they have to be very conscientious of having things like avocado, peanuts. You can't just say, oh, I just don't like those, I'm not having them. If you choose to have a vegan lifestyle, you have to consciously eat some of these higher fat, plant products so that your body gets enough. Does that make sense? Like that's one of the biggest issues I see with actually your age group making the decision to go vegetarian and vegan. If your family has not been vegetarian and vegan, is oftentimes I find at the teen years, you have people who are like, yeah, well, I like all of these vegetables. I'm good with these vegetarian meals. The problem is you can have those as your main, but you also will have to eat some of those vegetables and things you might not like because they have some of the stuff, like plants don't need cholesterol, right? They have cell walls for support. Our body doesn't have the cell walls. We have to have some cholesterol. Does that kind of make sense? All right. So guys, do we have all of the top filled in? All right. So what I'm going to have us do is I'm going to have you color these. I do want you to go ahead and have them color coded. So whatever for each structure, I want you to just be consistent. Now, I don't care what color that happens to be. You just want to be consistent with it. So for example, if I were to start, I grabbed a blue one first. If my phospholipid is going to have two colors. If I have the head, I picked dark blue. Then everywhere in my diagram where I have one of those little circles, I want to color it dark blue. Now, guys, you don't want to do E. E was a separate structure, so leave E uncolored. But you want to color all of the phospholipid heads, whatever your first color is. And it could be any color combination. You just want to be consistent with whatever it is. I'm going to do both the top diagram and the bottom diagram. So for example, this was all of the phospholipid heads. Yes, the tails are going to be a different color. I would do the tails a related color, but the tails we're going to do a different color. So it doesn't matter what color you pick. Color. But it was, I just colored all of the phospholipid heads one color. I colored the one color. That way it'll be nice and easy for you when you're looking, be like, wait, what is that thing? Oh, it's the purple one. Now, whatever color you pick, you do want to either circle or underline the name of it up top, just so that you remember what you colored that color. Yep. Well, because remember, in our body, anything that doesn't have the identification correctly, our body kills. That's where you're abusing. So if you're missing your ID, your white blood cells go around and eradicate. If you don't have an identification, your blood cells are going to be like, can't die.
Oh, yeah, they won't. He's going to his way.
You're part of that. And that, hey, we're good. Leave us alone. We're okay.
Or he brought it out.
Does that make sense? Like, not just that cell, but the cells around it. Maybe like, I don't know. You're trying. No, I think we might need to. The default setting in our bodies is if we don't recognize it, destroy it. Because that's the safer method, because there's so many pathogens out there, and they change all the time.
Hard enough. Hard enough.
So if at any point it's like,
Wasn't too-
we should sure about that. So this is why we have so many autoimmune disorders, is our own body basically.
too fast.
So now, guys, once you have all of the phospholipid heads colored, you want to pick a different color and do the phospholipid tails. So I was doing my one in a light blue. And that's the in-betweens, those tails. And it can be whatever your aesthetic is. You do not have to go. I just tend to go in rainbow order because that's my default setting when I color things. But if you prefer like all different shades of blue, as long as you have enough shades of blue, that works. Like I just. With the tails. Different color. So if you have a color that you would like that you're not seeing, I do have like the larger selection of colors. Like these are just the. I do have in the black cabinet there, I have basically all of the pencils and stuff that were out to work. I just sorted out the rough color category. Additional shades. I didn't have enough to have every problem people get. Then it's up there, not. Huh? Because it's color. Because it's the wrong thing. Like we had it. Like where it is on the top or at least. Like it's gonna have its own color when we color it. That was why I said, like, don't color it just like a regular phospholipid. It's a glycolipid. We're gonna put the colors. Yes. Those were the tails, right? Now, once you have all of your phospholipids, and go on to our proteins. So our B, we want to pick a color for B. I'm going to pick a light green here. Now for B, we have one up at the top, and then the two proteins on the bottom membrane are also channel proteins. Those two are also channel proteins at the bottom. Yeah.
When are we picking Lisa?
The MISA is in May. If you actually look at the calendar on Canvas, I do have it blocked out. I have the entire window blocked out. I don't know when our school is going to be administering it until we get closer. So I just blocked out the whole list of like possible dates. And then they're gonna tell us when we in particular are due to take that test. Because our testing coordinator, like that window is for the MISA, the park tests, the like all of the state tests. And then she tells us like each one gets a week. If that makes sense.
Yeah.
And she'll tell us when our, so. Sometimes like math and English go first, sometimes science and history go first. All right, so we want to color in our channel proteins and our signaling proteins. I did the channel proteins one color, signaling protein another color. I did light and dark green. A lot, there's always big. They're just small, but they only let. Here, we need to have energy. The signal would be like, hey, there's something happening, you might want to go on your protein channel. Anything, it tells the nucleus, it tells the cell membrane, it tells like whatever method has to go back and forth. Outside of the cell, inside of the cell. It might be the brain being like, hey, adrenal gland down here, come on. So the adrenal gland cells go, oh, come on, ER, come on, let's pump this stuff out and sending it out. It's like the PA system of that section. Like it gets the messages from the outside and into, and then the nucleus processes that information. Yeah. Yeah. This one's the same. Okay. These are all, yeah, they actually let stuff. Okay. Now, once you have your channel protein and signal protein, the next one you are going to want to look at is the glycoprotein and the glycolipid. You want to color those a certain color. And then the same thing for our peripheral protein and our cholesterol. No, there's only channel ones because the whole thing down here is just stuff moving and how it actually moves. And so I don't need to know signals, it's just what things move stuff. So now, once you have it all colored, this is roughly what we end up looking like. Any different colors, but this is roughly. Now, guys, the reason I had you color that bottom row as well is we want to be consistent of like what the different structures are. that bottom row, the bottom membrane, is not about the structures of the membrane. And so we've already gotten that part colored, and then we just have to fill in. Yep. I've been trying to start with a different part of the rainbow each one.
Two is 10:25 a.m.
Okay, so guys, I am. So we will do our video next class. I am going to have us jump to our exit questions. We're in the last 5 minutes here. What kind of transport, going back to your key ideas today, diffusion, facilitated diffusion, or active transport, which one of those do you think is going to react the fastest? Active transport, and guys, why do you think that's gonna? What do we use when we do active transport? Energy, right? When we put energy into things, it happens faster. Right? If we're just waiting for stuff to kind of happen, yeah, it might happen, but it might take it a minute, right? It's not always as quick. This makes sense? That's not reacts faster. The cell does stop it because it doesn't want to just burn up energy if we don't need it. But as far as the speed of something happening, having it be active transport, putting energy into it just makes it go fast. Whether or not, so usually we do active transport to make things more concentrated. Sometimes they'll use active transport to spread stuff out. But both ways, it's basically like turning on a pump versus just letting it do its own thing. Does that make sense? An example here would be if you've ever had like the inflatable like bouncy houses and stuff, you can have them go flat by like just turning the fan off and walking away, it'll go flat. Or you can turn the fan on reverse and then it like sucks the air out quickly. Same type of thing. As soon as we put energy in, it's gonna go quick. All right, so guys, next class we are going to fill in the words at the bottom and our front little edges. And then we'll watch our videos and talk a little bit about osmosis versus diffusion.